So I think there's always a question about whether vaccines still work in particularly old and frail populations. And we now have these new enhanced vaccines that are specifically for 65 and older against influenza. And it's one thing to say you have a more immunogenic vaccine. It's another thing to be able to say that it actually offers added clinical benefit, especially in the oldest old and the frailest frail. And that's the reason you'd want to study a nursing home population. I think if you can get a difference in an outcome in a nursing home population that's clinically important, that gives you hope that you would see it in all the other populations too. In the oldest old, they actually will have bigger signals. They have higher hospitalization rates. If they get sick with the flu, they're more likely to get pneumonia, have heart attacks, die. So it's easier to measure a statistical difference and also a clinically important difference in populations where the background event rate is already pretty high. In this study, we did this, it's called a cluster randomized trial. And what that means is rather than randomizing people, we randomized places, we randomized nursing homes. These nursing homes housed on average over 100 people per nursing home. And the value of doing that is that when you offer the vaccine, it's offered at a nursing home level in the same way they normally would do it, like their standard of care. So it's randomizing to two standards of care. The nuance of doing that is also that when you do a more traditional randomized control trial where you individually consent each person, those people typically have to be able to give consent. And that makes them different than the typical person who lives in a nursing home. So you'd be excluding most of the population that are quite frail. So if you want to have a generalizable result, you'd like to have it applied to the population the way you would in the real world. And so randomizing the nursing homes rather than people makes a lot of sense. So a second feature of the study is because this is offered as standard of care, when an individual vaccine is given, they have the right to refuse just the same way they would any season where they get offered influenza vaccine. So about 15% of the population in each group refuses vaccine, and that was the case here. And so you get to also extrapolate what happens at the whole nursing home level in terms of outcomes, not just to the people who get the vaccine. This is in part important because flu is a contagious disease, and this notion that if you get flu, you're more likely to spread it to the next person. So the more vaccine is around, the more sort of tailwind you get to a beneficial effect in the unvaccinated population because of that overall coverage. You know, we would traditionally call that herd immunity. And you can measure it if you're also including people who don't get the vaccine. So this is the top line pre-specified result was based on all-cause hospitalization. The overall study is actually powered on respiratory hospitalizations and pneumonia. So this is before we talk about just differences in those hospitalizations. So all-cause hospitalizations has the advantage that it's hard, especially when you do it at arm's length, to figure out when somebody has flu because we miss most of the cases clinically. In kids, you know, flu is clearly a respiratory disease and otitis and so forth. But in old adults, it can be heart attacks and strokes and other kinds of things that if you don't measure those, you would actually be measuring things that were consequences of flu that you would just never know about. So all-cause has the advantage that it's nonspecific and the advantage that it's all-inclusive. So our top-line result was for all-cause hospitalization in the pre-specified analysis was 6% fewer hospitalizations in the group that got the adjuvanted influenza vaccine. There's some nuance to that. Every flu season is different. This was an H3N2 predominant season. So that specific flu strain typically causes more severe disease. And we know that when we look at the immunology behind the adjuvanted flu vaccine, that in vitro, ex vivo, it performs better with AH3N2 than it does for other strains. So it's a place where we may be more likely to see a clinical benefit. Having said that, it doesn't mean it can't also give benefit to H1N1 or influenza B. And it also doesn't mean that this particular season was going to be the same size benefit you get on any other AH3N2 season. So part of that nuance is that in this particular season, 2016-2017, the overall efficacy of vaccine was considered to be pretty low for the season. And there is this, I think, relative consensus opinion that this was because vaccines that are produced from egg, so you inoculate eggs with the virus and then the virus that comes out is now an egg-produced vaccine, was a reason for a lot of that lack of effectiveness for vaccine overall for the season nationally. So the two vaccines we compared were both egg-based vaccines, and when you have this problem with vaccine effectiveness, it's called egg-based mutagenesis. So both of our vaccines were subject to that potential problem. So in the context of that, we still measured a 6% difference, and we don't know if that difference would have been larger if that egg-based issue had not been in the background. A second nuance is this is a large study. We did over 800 nursing homes, some 52,000 people in these homes that were part of the analytic sample. When you do this and you have that many facilities, you expect that all the features that are present in the two sets of nursing homes would balance themselves out. When you do this, they almost always do because this is called regression to the mean. But in our case, actually, there was a difference, an imbalance in African Americans between the two vaccination groups. Life happens. When you do a post hoc, so this is not pre-specified, but a post hoc adjustment, the standard error in the measurement actually goes up a little bit and the p-value drops. So we have virtually the same effect size for hospitalization, but we lose significance in the p-value because of that. So on the top line pre-specified result, it looks like there are fewer hospitalizations, but in the post hoc analyses that might be less. These are preliminary results, so we are hopeful that when we can analyze the sample for the other outcomes, pneumonia and respiratory hospitalizations, where the specificity to a flu vaccine effect would be greater, that we'll be able to show that those also hold up, in which case all of the results become more believable.